it's Peter here with my FSHD. All right, we've made it. You stuck with it this far? Did you see 1 and 2? I hope so. I know it's a lot, but you know what? It's important, okay? But now we're going to talk about the EPI-CRISPR-321 clinical trial, the design. There's no data, of course, but we have the design and we can infer some things. We're going to talk a little bit about how clinical trial is done. Now, make no mistake. This is an FDA-approved clinical trial. Don't get me wrong on that. This has gone in front of this IND, Investigational New Drug Application, has gone in front of the FDA. The FDA has given their stamp of approval to go ahead on the clinical trial design. They were convinced as much as they need to be about the preclinical data, but just so it's clear, we're giving our opinions, my opinion, Chris's opinion, on the science, our interpretations of the science, as having, for me, more than 20 years working on FSHD. For Chris, holy cow, but now it's almost 15 years. And also one of the world's leading experts on skeletal muscle gene regulation. And so, we're using our expertise, and these are our evaluations. But just, the FDA has determined this trial has passed all federal requirements for safety and for whatever the requirements are for potential benefit to patients based on whatever their requirement. Honestly, I don't fully know, because I thought I knew, but in that case, I can't understand how this trial got approved. But whatever, it did, it's there, and that's not any of our business, not being critical, just I'm not the only one with the same questions. Anyway, we'll bring everyone down with us. Anyway, we're going to go through the trial. As always, we're putting our necks out there. And again, I'm putting my professional reputation out there on the line. Okay, who else is doing that? All right. I actually have nothing to gain by this and everything to lose in some ways, because I know people are going to come after us. Okay, I'm doing this because it's important. This is our life's work, is to help people with FSHD through diagnostics, through helping companies develop proper therapeutics. That's why we made the mouse model and gave it away. That's why we made the pig model and given that away. That's why we've just published 2 other mouse models and given those away, right? That's why we help labs compete against us by helping other modalities, other companies who have therapeutics. We help them get the best science so that their technology can go. Let me tell you, there's some really good technology out there coming, and we're helping people that need help, because you know what, some companies really do want to get the right answer, right? They'll say they do. Some walk the walk, some don't. Some just talk, just like foundations. There are good foundations out there too. And make no mistake, I sometimes group everyone out there together, but we still, more than anything, appreciate Friends of FSH Research, Terry Kalila, always doing a good job. They're on your side. FSHD Global, Emma, Natalie, Lisa, they're on your side. Okay, there's good people out there. FSHD Canada, Neil Camarda, Craig Kelly, the gang, Fabio and Abreu, and there's some people on your side. They may not have the science chops to back it up on something like this, or may not even feel it's appropriate. Maybe that's not the job of a foundation. I don't know, you can decide. They're patient advocacy foundations. I understand it's a tough thing to pick sides. But you know what? That's why I'm here. We'll do the tough work. So let's go through the clinical trial. Let's see what we got. All right, Chris, you want to come back? All right, now here we are. We set you up. You understand the preclinical data. You understand the concepts. And now we're going to look at the EPI-CRISPR EPI-321 clinical trial design. Of course, there's no data here, but we're going to take all the data we know and we'll evaluate the potential for this trial to answer their questions and to learn anything about the system and any potential for benefit to participants in the trial. All right, so now we're going to talk about the clinical trial. Okay, we've done part 1 of this big. It turns out this is, part 1 was kind of the intro. Part 2, the meat of it was the preclinical data. Now part 3 here, we're looking at the clinical trial. So I pulled this from the FDA.gov website, directly word for word from the FDA, designing clinical trials. While preclinical research answers basic questions about a drug's safety, it is not a substitute for studies of ways the drug will interact with the human body. Clinical research refers to studies or trials that are done in people. Okay. Researchers design clinical trials to answer specific research questions related to a medical product. Drug developers must submit an investigational new drug IND application to FDA before beginning clinical research. In the IND application, developers must include animal study data and toxicity, side effects that cause great harm data, manufacturing information, clinical protocols, data from any prior human research, and information about the investigator. You also are going to include your preclinical studies suggesting efficacy, mode of action, and these things, but that's a little bit looser. But what I thought was most interesting was this last part. Even though the FDA offers extensive technical assistance, drug developers are not required to take FDA's suggestions. As long as clinical trials are thoughtfully designed, reflect what developers know about a product, safeguard participants, and otherwise meet federal standards, FDA allows wide latitude in clinical trial design. Okay, so I thought that was particularly interesting because what it says is they're not going to tell you how to do your trial. Right? They can advise you, hey, we got a lot of experience in this, and we have experts that can help you. But in the end, you get to do what you want to do as long as it's safe and meets certain standards.
Right.
But you can ignore years and years of experience if you want to.
Right.
Okay.
Yeah, that was interesting.
All right, a phase 1 trial, is now basically phase 1 is going to be safety and dosage, right? Phase 2 as well. But basically now, again, in gene therapy, right, the EPI-CRISPR trials, 9 participants. So gene therapy, because of cost and other safety issues, is much smaller. Right. The Fulcrum trial was more along these lines.
Yep.
But you're going to basically, typical phase 1 is healthy subjects. Phase 2 is those with the condition. So a phase 1/2 is dosage and safety with people with the condition. But you're going to learn a little bit about efficacy and side effects, right? While the primary outcome measures are typically, again, safety, and dose escalation, you do start to get answers as to efficacy.
Yep.
Okay, that's in phase 1/2. And then a phase 3 is really about efficacy, properly powered efficacy.
And long-term.
And long-term and monitoring
Problems,
adverse
yeah.
reactions, right? So phase 3 is where a lot of, you'll see here only 25 to 30% of drugs get through phase 3.
Yeah.
So phase 3 is the big cutoff because a lot of drugs are safe. A lot of drugs, if you look at the data through rose-colored glasses, like Fulcrum did, you might be able to convince yourself that there's efficacy, that it's pointing in the right direction. And if you write enough positive things about it and tell people that good things about it, that maybe in phase 3 it'll be good.
Yeah.
But more often than not, phase 3 fails.
Yep.
In fact, the fact that so many fail in phase 3 tells you that people,
You probably should have failed earlier.
they look at the efficacy data probably very biased way. If there's any indication this might be working, we're going to go.
Any statistical test, any bizarre functional study,
And then in phase 4, it's after FDA approval.
Yep.
Okay, it's after FDA approval. Now, an interesting thing I've also found on the FDA website is that they have a whole bunch of guidances. Look over here on the left if you're looking online, biological guidances, allergenics, blood guidances, tissue, vaccines, xenotransplantation, whatever. But they have a special guidance for cellular and gene therapy. Essentially, they took the experts in the field. So what do we know about gene therapy and put it all together so we can actually help you design a safe and effective gene therapy trial. But again,
you don't have
much to my dismay, it says, and I quote, in general, FDA's guidance documents, including this guidance, do not establish legally enforceable responsibilities. They can tell you what you should do. They can tell you what the responsible thing to do is, but they can't force you to do it unless it's not safe.
Right.
Instead, guidance has described the FDA's current thinking on a topic and should be viewed only as recommendations. I mean, it sounds like Jack Sparrow. It's just, they're really rules.
They're We're like loose recommendations.
Okay, viewed only as recommendations unless specific regulatory or statutory requirements are cited. The use of the word should in FDA's guidances means that something is suggested or recommended, but not required.
Well, this would explain why some ineffective therapies get through clinical trials.
Even though there are volumes of data available and experience and experts and tons of things, you can ignore it all and recreate the wheel if you are motivated to by whatever motivation you have, and plow forward with a predictably ineffective trial if you want to, as long as it's safe.
Yeah, interesting.
All right. Non sequitur. So, the EpiCRISPR trial, here we go. First in human study of EPI-321 and facioscapulohumeral muscular dystrophy, a phase 1/2 open label dose escalation study to evaluate the safety, tolerability, and biological activity of EPI-321, an AAV-rh74 delivered epigenetic editing therapy in adult FSHD patients. It's going to be Atlanta, Auckland, and Sydney. And I don't know anybody at these locations. I don't know the MDs involved, and it does not appear that there's a single FSHD. To me, I do not recognize a single FSHD expert at all. There is a worldwide FSHD clinical trial research network, CTRN, run by Jeff Statlin and Nick Johnson, that has the world's leading neurologists in FSHD with vast experience in running clinical trials, really in clinical, because they do myotonic dystrophy and LGMD as well, but they've done the FSHD clinical trials. They know how to assess efficacy. They know all the metrics. And these guys are just amazing researchers and clinicians, tremendous facilities, and EpiCRISPR is not going to use any of that. But that's their choice. They don't have to.
Yep.
I would make a different choice, but that is me. And again, we're not saying what's right or wrong. We're just saying my opinion is that's an odd choice. Directly from clinicaltrials.gov, brief summary. The goal of this clinical trial is to learn how safe and tolerable EPI-321 is, and whether there may be early signs it is working in male or female adult, 18 to 75 years, participants with FSHD type 1. The main questions it aims to answer are, how safe is EPI-321, and how well can people handle it over time? How does EPI-321 interact with its target, and does it show early signs of working? Okay, so those are the questions. Now, I like those questions.
All right, I'd like to know the answers before doing the
Well,
trial.
now wait a minute. Remember what they said. It is true that no matter how much dog and mouse and primate work you do, it's still not human.
Yes.
But you can get some indication.
How does the drug interact with its target? That is something that's addressable, in my opinion, in preclinical
So you
studies.
could actually, okay, but they've done some of the, okay, so let's just think about it. So what do we think about the questions? Whatever. The real question is, I'm thinking of this as a grant. Will the experimental design answer those questions? Ignore everything. Just say, will doing a single dose, which is all you can do, of 2 times 10 to the 13th of Rh74 EPI-321 tell you how safe this is? It'll tell you how safe that dose is.
Yeah.
Sure, so that will answer that this low dose is safe. Will it tell you how does it interact with its target and show early signs of working? I would make the argument that cannot answer that question.
Yeah.
Because
on everything we know.
Based on everything we've seen from their data and everything we know from other data, that there's not going to be enough expressed to answer this question, enough cells for a long enough time to answer this question.
I think we went over that and over that.
But that's my opinion. That's my opinion. If I had to predict it, I would predict, based on the primate data, that people will tolerate these 2 doses, either one, fine. If I had to guess, which I do have to guess, but I would also predict that they will learn nothing about the drug that's not already known. AAV, that's my prediction. Okay, so what do we got here? Participants will receive a single dose of EPI-321 through a vein while being closely watched in a hospital and visit the clinic regularly for tests and checkups for about 5 years. Really? I wonder if you have to. So I wonder if at three.
Standard.
So it's interesting. At 3 months.
Yeah.
So 3 months is 12 weeks, right? Basically.
Yep.
Dong Cheng Duan's data said at 12 weeks, all the CAS is gone. They're going to take a biopsy at 3 months. My prediction is all the CAS will be gone. They will see no expression of CAS, nothing.
Yeah.
Are you still stuck for 5 years?
I don't know.
If
Like,
hypothetically, if that were the answer,
right? Would you then not have?
Are you still stuck? Did you even take the 12-month biopsy, or is it already just trashed?
It's a good question.
I don't actually know,
idea.
because if you're hooked on for 5, now you can't get another AAV injection.
Right. In the meantime,
I don't know what'll
or
happen to the.
maybe ever.
Now again, that is a height that that's just based on data with other CASs and other systems with other things, and based on our interpretation of their own preclinical data in the monkeys.
Yeah.
And I've seen nothing that says that's not going on, but that's a potential thing. Okay. Detailed description. EPI-321 is an investigational drug comprising a recombinant adeno-associated viral vector serotype Rh74 for the delivery of genetic material encoding an epigenetic editor designed to address the root cause of FSHD. Again, I actually don't have a problem with the design. I have a problem with Rh74, but Rh74 has been shown to transduce human skeletal muscle efficiently in the clinical experience. I mean, so don't misconstrue efficiently with specifically. EPI-321's transgene product, a non-cutting nuclease-dead mini-clustered CRISPR with fuse epigenetic. Oh, come on! At least get the
grammar right.
At least get the grammar right. Christ, you're asking people to give muscle biopsies. You can't even type out a freaking.
And this is you.
I know exactly.
You're horrified.
Well, I will read it. With fuse epigenetic modulators, is designed to selectively bind the D4Z4 repeat region via the accompanying guide RNA, methylate CpG groups within the region near the DUX4 gene on chromosome 4q35, and thus repress the expression of toxic DUX4 protein, ameliorating the downstream pathology that drives FSHD. As it is under a muscle-specific promoter, which there's no evidence that suggests it is, the decastonics proteins is expected to be preferentially and actively expressed in muscle tissue following a single IV. Now, again, looking at the data that they presented, there's no reason to expect to be preferentially expressed in muscle tissue.
Certainly wasn't in their data.
Their data does not support their own detailed description.
Nope.
EPI-321-02. Clinical trial is an open-label dose ascending study of EPI-321 for safety and tolerability to determine the best dose for a future trial of drug activity. Now, this is confusing to me. I mean, I predict, it says the 2 dose levels will be evaluated, right? I predict that the outcome that is predicted by every other trial that's been done and their own data is that neither dose is worth it.
What are their benchmarks anyway? Do they say?
Well, safety. Remember, they're only looking at right, so but they are going to actually look at here. Okay, I guess I'll finish the statement.
Okay.
In addition, this study will collect secondary outcome data on muscle function, imaging characteristics, and other markers of disease activity at the baseline and throughout the study to assess their utility as measures of drug activity in the future clinical trial.
But how are they supposed to determine which is the best dose?
Well, so if you have 2, well, that's my question. So
you got 2
so
ineffective
you have
doses.
2 doses, neither of which even approaches an effective dose based on their data, in my opinion.
And both of which, in my opinion, are going to be safe.
So you have 2 safe doses, but you could say we are going to inject saline and determine what dose of saline is going to be effective for FSHD.
Yeah.
And you can say, we determined that saline is not effective for FSHD. I guess we need to do another clinical trial with a higher dose of saline.
Yeah,
I mean, that's, being facetious, but I don't understand because the implication is that one of these doses will be better than the other.
Right.
Now, if you look at, look at Fulcrum, they tried 2 doses and the answer was neither dose is good enough.
Right.
And that was it.
Yeah.
You look at Avidity, 2 doses, actually the lower dose was better because the higher dose gave you no benefit.
Right.
The lower dose is actually effective.
Yeah.
So that's cool.
That's useful.
That's useful. You're in the ballpark.
Right.
Now, they might say, we just need to do something to get in the ballpark. But their own data that we just went through in the preclinical data says they're not even in the city where the ballpark is.
That's right.
In my opinion,
Yeah.
in my interpretation of the data. I don't understand how you do this dose and think that the answer isn't going to be neither dose has any efficacy
so
whatsoever.
we have no idea where to go from
So
there.
now you have to do it again.
Right. No, that's crazy.
Now, if you're going to do it, so now, and it's funny. Now, people at the FSHD Society got to make it. When I say, "Oh, we learned nothing from a trial," oh, of course we learned something. We ran through a trial and we learned all these wonderful things from the Fulcrum trial. Wouldn't it have been nice if you had learned those things with a drug that had a chance of working? And predicted by phase 1/2. Well, sorry, preclinical data suggested it might work and was worth trying. The phase 1/2 data said it didn't work and there's no point in going to phase 3.
Right.
That was my interpretation of the data, which turned out to be correct.
Yeah.
So I look at this and you say, okay, so you got two, so what are you going to learn? You learn that you need a higher dose of RH74. Well, I think everybody kind of knows that.
Yep.
I think everybody, I don't know that that's not a fact that everybody knows that, but I am of the impression that that's well known in the field. In fact, they even had that in one of their things, that it is well known that high doses of AAV are required for efficient. So they seem to know that. And they tested that high dose in the safety profile. They seem to know that. I don't understand why, based on all the data, looking at all the data, they then said, oh, you know what, that's borderline safety, but we're going to get efficacy going 6 times less. I saw nothing that says that's true. In fact, everything I saw says that's not true.
Yep.
But again, maybe I'm an idiot. And I would love for somebody to come on and send me an email and tell me that I'm wrong about all of this and put your name behind it, though. Publicly when you're on Facebook and you're online,
You really would love that.
I love being told I'm wrong and being proven, because I learn. I love to learn. What have I missed? How have I misread this? My God, should I be looking at another profession? I might be anyway, but okay. So I don't understand what they're going to learn. Okay, eligibility criteria, whatever. So what are they going to do? Here's the design. You got 2 times 10 to the 13th viral genomes per kg, or 4 times 10 to the 13th viral genes per kg. These are the doses that they have chosen. 1.2 e to the 14th, right, was actually all the primate data, and that was safe. So we're expecting that you'll be fine because it was safe for 39 days. So that's fine. So safety's fine. Now let's look at doses of things that are out. Zolgensma for SMA. 1.1 times 10 to the 14th vigs per kg AV9. Elvidis for DMD, 1.33 times 10 to the 14th for RH74.
Yep.
Right? So you can go higher based on just the virus. Now, I don't know what the cargo does. Again, maybe they have data that we don't know about, but this is shockingly low.
But at those threshold doses, you are going to get a few deaths.
Yeah, it's a few deaths.
Well,
if it's
people don't think it's a problem. A few
until
deaths.
it happens
if
to
you
them.
and if you have an effective drug that works in most people, that's actually considered okay. But if you have a few deaths, in a drug with a drug that's not working, and everybody's already upset because the drug isn't working, that is not okay.
So we covered that.
from what happened.
We covered that in the previous podcast, right? That Zolgensma, there's a few deaths, but the drug works great.
The drug works great.
So they're
still out there.
accepted, but
It's accepted.
Elvidis has a few deaths, and the drug is
drug
kind of
never
really,
worked.
it's really questionable how well it
Very
works.
questionable,
Depends on who
and,
you're asking and how
yeah.
invested you are in the drug and the company.
Yeah, so it's a different metric.
So what are the so the question is, does the trial fail or succeed? Well, that depends on what your metrics are for success. So primary outcome measure, they're just looking for adverse events. So basically, it could actually meet the outcome measure. These 2 doses have no adverse events. Now the problem is, if they have no efficacy based on secondary things, which we don't expect them to have, I don't think you can then suddenly double the dose.
That's what I was going to ask.
Don't you have to do the phase 1 again and look for adverse events again? So now you have to go back and do it.
Right. So you have to choose one of those doses to go forward.
Or somebody says,
double it.
you guys are idiots for using RH74. Why don't you use a myotropic vector?
Well, then you have to redo the
Now you have to redo it again. Now you have to redo everything again too. So in my opinion, the predictable outcome is there will be no adverse events. but there will be absolutely no efficacy.
Yeah, and they'll say, well, that's what we expected because
So-
this is just a phase 1 trial.
So they're going to look down here and say change in vector, but it's phase 1/2. In phase 1/2, you are starting to get indications of efficacy. And they actually said they want indications of efficacy.
No, they did, yeah.
So change in vector copy number within skeletal muscles. Yeah, can you go lower than 0? You know, cargo transcriptional activity within skeletal muscle biopsies, baseline 3 and 12 months.
Okay.
DUX4 expression,
they're gonna
change
look
downstream
good.
marking DUX4 and skeletal, and methylation status. Now the thing about this, so they could have gotten the first 2 basically in primates, right? They could have looked at copy number at 3 months, and that'll tell you if they're getting durability and activity, right? So they could have looked at biodistribution and activity. Now, the second 2, DUX4 expression methylation, are going to go alive. If you don't have the activity, you're not going to get that. So in some ways, you don't need it. So if these are positive and you have tons of vector and tons of cargo activity, you can infer that you're going to repress DUX4 and based on their preclinical data.
Right.
But if you don't get good high copy number,
no
remember,
way.
they needed 12 copies per cell.
Yep.
And all the math says that they're 50 times lower than that.
Yep.
Transcriptional activity, the data suggests that the cells are being cleared, and the data suggests from other systems that the cells are going to be cleared.
Yeah.
And at 3 months, you'll know this. So you could actually know this is a go, no go. Right? You do this at 3 months. If the answer is, wow, we don't have very much in there, and boy, all the cells are gone, you would never go to clinic. But again, that's my opinion. Right? I think that, to me, if it was me and I were doing this, I would have the answers to that before going to. Now, maybe they do. Maybe they have those answers and they just aren't telling anybody. I saw the primate design. They sacrificed the monkeys at 39 days. They would have to have done an entirely another primate experiment in order to do this because the monkeys were sacrificed.
See.
Well, I'm just saying that there's no indication that they have done the work.
I agree.
But maybe they have. We actually don't know. Okay, so here's the clinic questions, comments, and concerns. This is me, my concerns. If I were writing to the editor of the journal that submitted this paper, or if I was responding to the grantee, or talking to the grad student that is going to have to redo their preliminary exam, I would expect a higher dose for FSHD as a gain of function than for DMD as a loss of function. I would expect that you need to hit more cells because the bricks can explode, where in DMD, if you put a little bit back, that can actually spread throughout the myofiber. So if you have one nucleus in the myofiber in DMD, it can spread throughout the myofiber, the dystrophin. You hit one nucleus in FSHD, you haven't methylated any of the other nuclei because you got to get it out into those other nuclei. And we know that things don't travel that much in muscle cells, right? They have little kind of these little myo neighborhoods.
I can see gradients of expression of things.
Okay, I would also, my concern, strikingly low clinical trial dosing in comparison with DMD microdystrophin, right? The biodistribution was strikingly bad in their NHP data. They showed with prednisone, they're getting about 1 vector genome per cell, or even less, actually, at their 1.2. Well, that's their 1.2. That's not even that's not even their dose, right? That's in their safety study, right? So at 6 times the clinical dose, they're getting 1 vector genome per cell. The myoblast data says they need 12, and so this really is a concern.
Yep.
While there's safety data, I don't see any efficacy data. Data in dogs and humans using AAV-RH74, albeit with other cargos, do not support these as being effective doses. Now, again, this is my integration of the data. I don't see this being effective. Durability studies on other cases find an immune response.
Yep.
Is there any data to support durability? Because I got to tell you, their data isn't very convincing. I don't care about the mouse data, but the NHP data is not convincing.
Yep.
And I'm going to go back to what Amber said. I'm sure she's a fantastic CEO, right? Engaging with the FDA for a new drug modality requires a bold, evidence-based approach. salesman advises biotechs, follow the data, follow the science, and advocate for scientifically sound results. We are taking Amber's advice right here. That is what we are doing. We're looking at the data, we're following the data, we're following the science, and we're advocating, meaning, I can't believe this trial is going forward, my opinion. That's my response, is I can't believe this trial got approved, and I can't believe that they're doing it.
Yep.
That's my feeling about it.
Yeah, no, I feel the same way.
Following what she says. Okay, so what did we find? We find they actually have dosed somebody August 5th. They dosed first in human clinical trial into a person, so this is going forward. I actually wasn't sure it was ever going to actually go forward. After I looked at this, I said, surely this isn't going to go forward. And I was wrong. See, I'm wrong sometimes. I was like,
You're
okay.
just too optimistic.
And so, okay, so what do we see? F21 is the first investigational gene therapy. Yep. FDA has granted fast track rare pediatric disease and orphan drug. So cool. So if it works, there's a lot going for it. Cool. What do they say? This is their press release. For too long, individuals with FSHD have endured this progressive disease with no treatment options, said Amber Saltzman, PhD, Chief Executive Officer, EPI-CRISPR Biotechnologies. EPI-321 represents the start of a new chapter where we're finally evaluating a therapy that targets the underlying cause of FSHD. We're thrilled to dose our first patient, I bet, which is a milestone for our team, and more importantly, a sign of hope for patients and families waiting for meaningful treatment options for this progressive neuromuscular disorder. Well, they're a company. They're responsive to their shareholders. That's exactly the type of response, right?
Yep.
That they're saying, wow,
Very par for the course.
I don't agree with it, in that I have concerns again that the hope is quite not supported, maybe false. But there's others. Certain, okay, FSHD Society, they have a quote. Well, this is good because now this is a patient advocacy group. Now the company is responsive to their stockholders, and I guess they're not public, but they have investors.
Right.
Push comes to shove, what's best for the company, for the investors? I mean, I'm not a good businessman, but that's my imaginative. Society, though, they're responsive to the patients. And they have scientists. We told you that. Lucian Ronco, Michelle Melian, right? They have a science advisory board. Certainly, they have looked at the science the way we have looked at the science, right? I wonder if they agree with us. Mark's never one for hyperbole. Let's see what Mark says. This is a deeply significant moment for the FSHD community. I'd agree with that, but I'm willing to bet for different reasons, said Mark Stone, CEO of the FSHD Society. For decades, patients and families have carried the burden of a disease with no treatment that addresses its root cause. That is true. I agree with that. They've shown resilience, courage, and unwavering hope. Well, you can raise a lot of money off of unwavering hope. Yeah. The dosing of the first patient with EPI-321 represents more than scientific progress. I agree with that too, but probably for a different way. It represents a turning point in lives, I'd agree with that, for those who have waited far too long. We commend EPI-CRISPR for advancing a bold and innovative approach that brings new possibilities and real hope to families around the world. I gotta say I disagree with that last one. That could have been right if the preclinical data was different, if the virus, in my opinion, if the virus was different, if the promoter was different, and if there was an indication the cargo actually worked on the target tissue, that could be correct with the right dose, in my opinion. I don't see anything that supports that. Why would a patient advocacy organization that's got a bunch of scientists associated with it and working for it and all about the science tell families there's real hope around it? Maybe, I mean, it could be that we're just completely wrong.
Yeah, let's hope so.
Well, I mean, I'm granted, like I said, they have some outstanding scientists there, and I hope to hear from them and tell me how I'm wrong and how they interpret this data differently. So we'll assume that we're wrong and that, okay, so, but I gotta tell you, I was looking at the website, and it turns out that this patient advocacy organization is actually a corporate partner with EPI-CRISPR biotechnologies.
I wonder their mission statement sounds so similar.
Wow, it's almost, I noticed that too. I noticed that it
could have been
sounded very, it's
interchangeable.
almost like they like work together.
Yeah.
Now we don't know that happened. We have no idea. And I don't know what being a corporate partner means, but it suggests to me, these are the corporate partners, that this patient advocacy organization might not necessarily only be advocating for patients. They actually, their intentions are, yes, advocate for the patient, but what happens when the company needs, I don't know, maybe they're always aligned, and that's the great thing. I don't know, but you got to think, gee, I understand that Amber's statement. She's a company person, great
Right.
CEO,
It's a business.
doing business. Yeah.
Yeah.
Everybody,
Talk the talk.
I get the feeling that, the company, I like the company. I like the platform technology. Stanley Chi is an outstanding scientist. No doubts about that as a CRISPR scientist. I don't know any FSHD person associated with that. I'm willing to bet that this company is going to be a wonderful success. Champagne and caviar for all the investors. Everything's great. But not the FSHD part. As for efficacy, I haven't seen anything that says this is good for the FSHD patients. I get the feeling, they're not getting champagne and caviar. I hate to say, I don't know. I'm a little. I don't know what Marks get. They're corporate partners. They're getting something. I don't know. I have no idea what the arrangement is or what that buys them. It looks like it buys them good press.
Yeah.
Remember when Fulcrum's disastrous phase 1/2 data was better than expected results, and they pumped everybody up to say yes, get in there.
Yep.
Boy, it sounds like it's not real unbiased advocating for patients, in my opinion. But again, maybe there's I can't believe they would put out a statement so supportive of a clinical trial when they wouldn't recommend that trial to their constituents. I would love to hear from y'all and tell me. Okay, and remember, everybody, okay. What does Solve FSHD say? Well, actually, they don't say anything. They're a major funder, and they don't say anything. But that's cool. Actually, I got no problem with Solve FSHD. They're venture philanthropy. Their job is to invest in companies and help them get over the line. Now, they have some scientists that are FSHD guys as well, who I know, who are pretty good scientists, but they're not gene therapy guys. I could understand if EPI-CRISPR came and said, hey, this is our platform. Can you invest in us for FSHD? I would probably invest, actually, based on the platform, the idea, these mini Cases and epigenetic regulation. Great. Yeah. But I got to tell you, I probably wouldn't put out a statement either if I saw that preclinical data and this was the clinical trial that I was financially supporting.
Yep.
That's me, though. I wouldn't want my name attached to this. All right, FSHD Global. Emma's always, you can always count on Emma. What's Emma say? Is Emma going to be a cheerleader or downer? What she got? FSHD Global, a disease-modifying therapy that halts progression, would bring our patients and families an improved quality of life and hope for a future without the burden of FSHD. Absolutely, that is absolutely true, Emma. We welcome this significant progress towards that reality. Says Emma Weatherly, CEO of FSHD Global. Okay, that's about as vanilla as it gets. That is, I think what you're saying there is what you're not saying. I will say, Emma, good job.
Yep.
Good job, Emma. I understand you can't come out and say certain things. It's just like writing a letter of recommendation. If you're the best student I've ever had, I'll tell you, Current phase 1 trial will carefully evaluate safety and tolerability with initial data expected in early 2026. While this timeline requires patience, it represents a methodical approach to ensuring any future treatment is both safe and effective for the FSHD community. That is accurate. I actually appreciate Emma's statement on that, right? I understand that. There you go. Good job, Emma. Always got it. Always got it. That's why she wins awards. So one thing haven't heard from, though, and I've never heard, in all the times I've seen Amber Saltzman talk multiple times, I've never seen an FSHD expert discuss this. Now, I was a KOL, key opinion leader, for Fulcrum. I went to New York City and talked to investors prior to their phase 1/2, and talked about epigenetics and the disease. Every company, they have KOLs, key opinion leaders, who tell everybody how great the technology is. I'd be curious who the KOLs are that are supporting this. I haven't seen, now I understand a lot of people don't like to have their name and their face behind anything when they make controversial comments.
Yes, everybody except for you.
Well, but everybody, people don't mind having their, everyone says positive things. No one likes to have their face behind something
Of
negative.
course not. Yeah.
So certainly someone must be out there, right? Where are the neurologists that are FSHD experts? Where are the FSHD researchers? Where's all these people saying that are experts in any aspect of this saying this is great?
No,
I haven't
very
seen
silent.
it. I couldn't find anybody. Well, that's not true. I found one dumbass willing to actually go on the record with a statement. We showed almost 10 years ago that CRISPR inhibition is a powerful gene therapy technology for repressing FSHD pathogenic gene expression, and FSHD is ideally suited for this type of gene therapy approach. However, translating to the clinic in a way meaningful for patients takes time, and the devil truly is in the details. As we have seen with other neuromuscular disease trials and extensively in preclinical work, there are narrow margins between safety, efficacy, and lack of benefit long-term, or even short-term. Any therapy needs to be both safe and efficacious. Fortunately, the gene therapy space is moving quickly, with many companies learning from past failures and limited successes, such that the industry is now equipped with new and better viral delivery platforms and muscle gene regulatory cassettes for safer and more effective NMD gene therapy. In fact, many are available now. In addition, there are now many tools available to FSHD researchers and industry for those who wish to utilize them to go to clinic with the best possible predictive data for safety, dosing, and efficacy. Unfortunately, EpiCRISPR Bio has chosen to forego these advances and tools in favor of old, less effective technology. In addition, I find the preclinical data lacking in many respects. As being witnessed with the recent DMD gene therapies, in the long run, patients will benefit most from going forward with gene therapies that were developed taking advantage of the best available science and tools and technology, and with the most rigorous preclinical data. While time is muscle, wasted time is wasted muscle. The words that most aptly come to mind are the mantra, if you want to get there fast, go the long way. Overall, in my opinion, I find the preclinical data to be weak and unconvincing, and the clinical trial dosing unlikely to provide any benefit. However, I do wish the trial participants the best, and I continue to look forward to a future with safe and effective gene therapies for FSHD. So there you have it. That's the short version. Now, I apologize for the length. It is quite long. But I hope you appreciate it's important. This is not a game to us. It shouldn't be a game to anybody.
Yeah.
It's not about money. It's not about finance. It's not about prestige. It's not about being first. It's about helping you and your family and your loved ones and our loved ones. Okay? Someone has to advocate for you, right? Hopefully you've learned a bit about gene therapy and the EpiCRISPR biotechnologies, EPI-321. You have to think critically. Okay, scientists, that's our job, is to think critically, to ask questions. And it's also our job to answer the questions that are asked of us. We don't get upset, we don't sue, we don't get, someone says, this doesn't look right, you explain it, or you do an experiment to address the issue, right? That's how science works. It's a back and forth, constant peer review, constant improvement, because there is a right answer in science. It's not an opinion, right? Now, what we shared our opinions and our interpretations of the data, but the data is the data. And Seinfeld did do a routine of that. And I just have concerns for the potential for this. I think this technology has great potential. I gotta be very careful what I say. I'm concerned that the potential for this technology to help FSHD is not being realized with the current design of the cargo and the current delivery by the virus. What if this was actually something that could really work? Granted, I'd like to see some data that shows it, but the last thing we want is to lose a viable therapy because of piss poor science and rushing to clinic. Now, I'm not saying they're rushing to clinic, but in my interpretation, the science is piss poor. That's my opinion. Anyway, Geiger, man, take us on home.
Thank you for listening to the MyFSHD podcast, where we share with you the latest news and information on FSHD as we strive for a cure. Do you have a question for Peter? He'd love to hear from you. You can reach him at peterjones@med.unr.edu. Thank you for listening, and join us again next time for more news and information.